Welcome to the screencast, where we will be demonstrating Cellenium, our platform for collecting and analyzing single-cell sequencing data, including scRNA-seq, site-seq, and ATAC-seq data. Our primary goal for Cellenium was to provide users with a high user experience, fast responses to user requests, scalability in hosting a large number of studies, system simplicity leading to small maintenance effort, and low runtime costs. Our internal installation hosts approximately 600 studies from both internal and external sources. Cellenium has 3 different modes. The first focuses on single study analysis, where you can analyze individual studies in depth. Cross-study expression comparisons can be done in the second mode, and in the third mode, users can potentially find cell types in which a gene of interest is specifically expressed. You can switch between the modes using the buttons in the top right corner. Let's begin with the search function. Integrated studies are tagged using standardized terms from ontologies, and we offer a semantic search that considers available synonyms. For example, you can type in species such as human, tissues such as lung, cell types such as T cells, or diseases such as COVID-19. We also provide an ontology browser where you can find studies of interest using the corresponding ontology trees. For example, from the NCIT tissue ontology. For clarity, we are only showing the parts of the ontology trees for which integrated studies are actually existing. The search result cards contain information such as the title, description, tagged ontology terms, and the number of cells in a study. Let's inspect the integrated pancreas study. You can see the sub-analysis sections on the left-hand side. We offer cell type marker gene exploration, expression analysis, co-expression analysis, interactive user cell type annotation, and annotation comparison. In the cell type marker gene exploration section, you can explore the cell types of a study by switching between available annotation groups. If differential markers for an annotation group have been calculated by the study integrator, it is indicated by the calculator icon. You can hover over the cell types and the corresponding cells are highlighted. You can also hover over the cells and the corresponding cell type annotation is highlighted on the left side. If you click on a cell type, for example the beta cells, you get the differentially expressed genes on the right-hand side immediately. By clicking on the eye icon next to the gene, you can quickly overlay the corresponding expression values of the gene onto the selected cells. If you want to do further analysis with a gene of interest in another section, click on the plus icon to add the gene to the user store. You can add and analyze any gene of interest by typing it into the input bar, for example CFTR. In the expression analysis section, you can generate violin plots, projection plots, and dot plots for any of the genes. You can switch between available annotations for each of the plots. If you want to filter out cells for some reason, you can do that using the filter out functionality. For example, let's regenerate the plot by removing the cells coming from CellSec or CellSec2 protocols. In the co-expression analysis section, you can generate pairwise scatter plots between selected genes. Again, you can filter out cells using the filter out functionality. You can also find co-expressed genes for any gene of interest by clicking on the calculator icon next to a gene, which triggers a transcriptome-wide correlation analysis. In the user annotation mode, you can assign cell types based on your own selections. Let's, for example, add the cystic fibrosis transmembrane conductance regulator CFTR and the matrix metallopeptidase MMP7. You can select the corresponding cells in the generated scatter plot using the lasso function. You can refine your selection further by adding another plot and typing in another gene of interest. When you're satisfied with your selection, you can store it, and in the background, differentially expressed genes are generated and can be explored in the cell marker gene section. In the annotation comparison section, you can analyze how different annotation groups relate to each other. Let's now go back and have a quick look at the cross-study analysis. Just type in your gene of interest and a cross-study dot plot will be generated. This is possible thanks to aligned cell types predicted using the DuiLab tool Cello. In the demo installation, we have only integrated a few public studies, but when many studies are integrated, you can filter using the search bar or the ontology browser below, in the same way as on the first study search page. Finally, we have implemented a marker gene search. Here you can possibly find cell types in which your gene of interest is specifically expressed within a study. Let's type in adenylate cyclase ADCY3, and indeed we are finding cells that are specifically expressing the gene. You can click and you will be forwarded to the selected study with the cell type and gene being pre-selected. Lastly, we would like to mention that Cellenium can also deal with site-seq and ataxic data. Let's quickly select the ataxic study of the demo installation. You can perform essentially the same sort of analysis. In the differential expression table, you will find differentially accessible regions, and by clicking on expand you can find out about corresponding genes and, for example, analyze their expression further. This is similar for site-seq studies, where you can find protein expression markers and also inspect the corresponding genes. With this, we would like to end our little demonstration and hope that you find our platform useful. Goodbye.